that surfactant deficiency and respiratory distress is at the same time that you get an acute inflammatory response. And this is an accumulation of neutrophils initially and macrophages thereafter. There's a further elaboration of growth factors and cytokines, which then subsequently causes fibrosis. So pulmonary surfactant is a gamish of different products, but it's really a phospholipid-rich material that includes distinct proteins. There's actually 16 different proteins, 4 of which have been characterized to quite a lot of depth. Surfactant proteins A, B, C, and D. And this is a very interesting substance which allows us to breathe normally right now. If we didn't have surfactant, we'd all be on ventilators dying. And it has many functions. It allows us to keep our lungs inflated at the end of expiration, so we don't have to keep inflating our lungs completely. It has antimicrobial defense properties. So surfactant proteins A and D are called collectins. They switch on macrophages to do bad things to bugs. And it's also involved in this recirculation of surfactant, if you will. It's evolutionary preserved. If you look at lungfish that only occasionally fly out of the water, they have the same surfactant that we do. So in thinking about models that I could study in terms of acute lung injury in adults and survivable models, hyperoxia and other models that were available usually resulted in the death of the animal. So you could not have a long-term view on how to study this. So I picked the intratracheal bleomycin model, which is a model of acute lung injury and pulmonary fibrosis, but it does involve oxidative nitrogen stress. It does have an inflammatory response where TGF-beta and TNF-alpha are increased around 7 days after the intratracheal dose, and then fibrosis develops thereafter from 7 to 28 days, and there's a slow resolution. So now I could at least look at the whole system and see how this interplayed. So the first hypothesis we came up with was that the respiratory distress that follows bleomycin injury would be a deficiency of specific surfactant components. And this is all published work, so I'm only going to show you one set of data from these studies. We looked at the respiratory rate, and this was done in rats, actually. So this one slide represents about 4 months worth of work where we sat and actually counted how fast these rats were breathing. And it was before the time you could get, where I could afford plethysmography. We've now got plethysmography systems that can measure mouse breathing rates, etc. But you can tell that the animals that were either uninjured or got intratracheal saline, there was no increase in respiratory rate. But the ones that got intratracheal bleomycin, as early as 2 days and maximally at 7 days, had increased respiratory rates. They were cowering in one area of the cage, they weren't moving, and they were pretty sick. And when we looked at the surfactant protein system in these animals, we found something interesting, that the SPA and the SPD expression was increased, whereas the SPB expression was decreased. It's interesting to note that because surfactant protein B is the singular protein that gives surfactant its ability to reduce surface tension to allow us to keep our lungs open at the end of expiration. And if you don't have surfactant protein B, your lungs collapse and you die. So there's a specific deficiency of SPB and actually SPC as well after bleomycin injury. So I thought that was interesting, and that'll become evident why that's important in a little bit. Okay. So I have been studying hyaluronan for 25 years now. It's a very simple molecule. It's glucuronic acid and N-acetylglucosamine, 2 sugars in tandem, and you can get small fragments and you can get large fragments. It's really a sugar polymer. And you would think that a molecule that simple would be easy, and unfortunately it isn't. It's evolutionary, that structure is evolutionary preserved in the sense that the bacterial coats that protect bacteria and impart virulence has the same HA that we have in our bodies today. So there must be something critical about this molecule that has preserved it intact for so long. But it's a glycosaminoglycan, it's non-sulfated, and it occurs in several different sizes. The high molecular weight is greater than 500 kilodalton, low molecular weight is less than 500, and the oligo HA is really less than 50 disaccharide units. And while high molecular weight provides structural integrity, I always like to say babies are cuddly because they have a huge amount of high molecular weight HA. And as we sit in the sun on the beach and destroy our HA in the skin or get older, we now get wrinkled. So I'm really 75 and I use my HA cream every day.
It's working.
It wouldn't be so good if I was lying. I wasn't lying. But there's a lot of products out there that have a high amount of hyaluronic acid. So every cosmetic that you ladies are using has HA and placental extracts, which are high in HA content. But when you get low molecular weight HA, now you start to get interest in biology, because now the low molecular weight HA interacts with receptors, which regulate adhesion, differentiation, proliferation, and motility. So what's known about HA in lung injury? We know that in human lung diseases, farmer's lung, sarcoidosis, ARDS, HA in the lung goes up dramatically, and it goes up dramatically early in the disease process. And as I'll show you a little bit in the animal models, multiple animal models have also shown increases in HA with lung injury. And herein lies a conundrum for HA and lung injury, because inflammatory growth factors such as TGF-beta increase HA synthesis. And yet, low molecular weight HA, when you put that on macrophages or other cells, now you can induce the production of cytokines, growth factors, including inos. So what comes first? Is HA the chicken or the egg? Okay? And that was sort of a perplexing question to me about 20, 25 years ago. And we think hopefully I show you that we've made some headway on that. So to make the biology interesting, there's binding partners and receptors that HA interacts with. There are cell-associated receptors. The transmembrane protein is CD44, is the sort of classic example of a transmembrane protein. Non-transmembrane protein, of which RHAMM, or receptor for HA-mediated motility, is a sort of example of that. And then binding partners that reside in the extracellular matrix, which almost sequester the HA and provide structure. And then there's soluble proteins that are circulating in our blood, like inter-alpha inhibitor. So there's a number of partners that HA interacts with. And then over the years, my first project in HA was to identify the HA binding site of RHAMM. Being a clinician, that was fairly esoteric, and I was like scratching my head as to what the hell am I doing here. But I'm glad I persevered, because I ended up defining a charge motif identified by BX7B, which was two basic charges flanking 7 non-acidic residues, which was the sort of basic HA binding motif. It's very similar to the heparin binding motif, which is BBXS, BBXS. So we thought that this was a good thing, and we could predict proteins that would bind to HA. We know that the link module in CD44 and other major HA receptors and binding proteins is a very important 3D conformational binding motif for HA. And inter-alpha inhibitor singularly is covalently, it's the only covalently bound protein to HA. And there's a huge biology with that in terms of how it interacts in sepsis and in other inflammatory disorders. So we were intrigued by this BX7B motif, and we generated peptides to figure out whether we could create something that could bind HA. And so what we did was we used arginine and glycine to generate the BX7B motif, and then added increasing arginine residues to increase the positive charge of this peptide. And my job was to put this peptide onto macrophages and measure the motility. And to my surprise, as we increased the basic charge of this peptide, we had increasing HA binding capacity, and an inhibition of cell motility. And there, sort of the light bulb went on, and I thought, put my clinician hat on, this might be a great anti-inflammatory agent. And so that's where sort of literally the lung injury project started in Eva Turley's lab. And I'm deeply indebted to her for not only introducing me to this exciting field, but being a mentor that I still work with and I publish with and just applied for a grant with 25 years after the fact. So that's for the junior people in the audience, it's really an incredible journey that you can take. So when we looked at the bleomycin model and looked at the HA system in the bleomycin model, the ED1 positive cells are the macrophages in the rat, and the saline ones did have a small inflammatory response, but nothing too dramatic. But between days 4 and 7, there was a massive 4-fold increase in the accumulation of macrophages in the bleomycin model. And this mirrored very much a 40-fold increase in the amount of HA that you could get out of the lung. And all of this was low molecular weight HA. So here in the bleomycin model, we can show that there's an increase in HA. It's low molecular weight HA, predicted to interact with receptors, and that this was involved at the same time that there was an inflammatory response. So the hypothesis too, since by this time I was indoctrinated into HA, thinking the HA is the center of the universe, which I still believe, that shoved down people's throats at UT Southwestern every day. And so in trying to answer that conundrum, what comes first, is it HA or the growth factors? Our hypothesis was that HA was the critical thing that mediated the inflammatory response, surfactant deficiency, and pulmonary fibrosis. So to put that into a model, we said lung injury causes HA, or low molecular HA, causes inflammation and then respiratory distress. And that we now had an HA binding peptide, the one with the most basic charge, which subsequently has been named NPI 0106, for God knows why, but so we're working with a pharmaceutical company startup to try and develop this technology as a potential therapeutic. So now we could answer the question of whether HA preceded the inflammatory response. And so what happened was that we did the same experiment over. We gave bleomycin, and without bleomycin, as we've seen before, there was an increase in respiratory rate at day 7. You had a scrambled peptide which had the same number of R and G residues, but arranged in a non-HA binding sequence, and there was no effect on the respiratory distress. And when you gave this peptide to the animals, you now had complete resolution of the respiratory distress. These animals were happy, scurrying around in the cage. They had no distress. It was remarkable.
Rashmin, when do you give the peptide?
So, sorry, that's a good point. So we give the peptide right at the time of injury. So intratracheal bleomycin was given. At the same time, we gave a subcutaneous dose of the peptide. I did not want I wanted it systemic, and I wanted to stop the influx of inflammatory cells. And I also didn't want the criticism that it's just binding the bleomycin. So I had to give it in a few different sites. And I sort of argued for 2 months with Eva about doing that. She wanted to give it intratracheally. So I went ahead and did the experiment, spent $5,000, and I prayed to God that it worked, otherwise my career in HA was going to be pretty short.
So it's just one shot?
One shot. And then 7 days later you have effects, and I'll show you 14 days later you also have effects. So the respiratory distress was non-evident when you gave this peptide. It was amazing. And so we measured the minimum surface tension of the surfactant from a lavage sample. And you can measure that using a bubble surfactometer, which actually measures the surface tension in this bubble directly. And so when you give bleomycin, the surface tension goes up. In other words, it takes a lot more pressure to open that bubble. If you give scrambled peptide, you still have surfactant deficiency with increased surface tension. And when you give the peptide, your surface tension was back down to normal, and that's why these animals were not in any distress. So when we looked at the expression of surfactant protein C, we saw decreases in bleomycin-injured animals. The scrambled peptide also had a decrease in, this is a northern blot, and with the peptide, essentially the surfactant protein C was protected from the decrease due to bleomycin. So this is truly remarkable. So why did this happen? I mean, were we affecting cytokines? Were we affecting the macrophages? So we actually looked at the ED1 positive cells again. We saw an increase with bleomycin. Scrambled peptides still had the increase in macrophage influx, and we had about a 60-70% decrease in the macrophage influx after bleomycin, suggesting that perhaps the way this was working was preventing the inflammatory response and hopefully decreasing the inflammatory cascade, the growth factor cytokines that were decreasing the expression, gene expression of the surfactant protein. So then we measured active TGF-beta in the lavage, and sure enough, with bleomycin, there was TGF-beta increased, as was known before. Scrambled peptide had also an increase, but there was no increase at all in the peptide-treated animals given bleomycin. So it was really incredible to watch this, these animals do so well. So we also had look at fibrosis. If your TGF-beta is not there, then you should have an effect on fibrosis. This is your normal lung. It shows a nice distal complex structure with alveolar intricacy. This is your lung on bleomycin. It shows areas of hyperinflation. This is trichrome stain, and it's fibrotic. This is the lung on bleomycin and scrambled peptide, the same overinflation and fibrotic lung, essentially a destruction of the, I'm in a room full of pathologists, I have to be careful. And here's the lung given the bleomycin with the peptide, and you can see that there's an almost complete abrogation of the fibrosis in this model. We also looked at collagen 1 expression as well as hydroxyproline, and those two were decreased. But it took me a couple of years to figure this out, but this is not a normal lung. If you compare these two, there is no protection of the alveolar component of this injury. And that's what my second half of my talk will be focused on, because this is what was the impetus for us to look at alveolar development. So since TGF-beta does transcriptionally regulate surfactant protein B expression, we had mice that were expressing a TGF-beta responsive 1 kilobase promoter fragment that had previously been shown to be responsive to transcriptional regulation through a transcription factor called TTF1 or NKX 2.1. And it binds to that site and increases the expression of SPB. And we knew that if you gave TGF-beta, the binding of TTF1 to that site was decreased, and so we generated mice that had this promoter construct driving a chloramphenicol acetyltransferase reporter. So here's a mouse with a cat, and we looked at now with enough money to buy a plethysmography system. We looked at the respiratory rate of these animals, and this is percent of control. If you give bleomycin, there's a 25% increase in respiratory rates. Mice breathe extremely fast, we didn't know that. The scrambled peptide animals also had this increase, and the peptide-treated animals had normal respiratory rates. We could reproduce the rat studies in the mouse, and when we looked at the chloramphenicol acetyltransferase activity, we could see that bleomycin decreased the promoter activity. Scrambled peptide had no effect, and there was a normalization of the promoter activity in these animals when they were given peptide. So the first summary for the piece that I've told you so far is that bleomycin injury with this HA binding peptide strategy decreases respiratory distress, decreased surfactant dysfunction, prevented SPB&C deficiency, decreased macrophage accumulation, decreased lavage content of active TGF-beta, and also decreased fibrosis at 14 days. So we think it's a pretty important finding. So how does this all happen? HA is the ligand So it must be interacting with receptors to mediate cell behavior that controls this process. So the two receptors that we've been interested in is CD44, which is the transmembrane type 1 receptor, and RHAMM, which is receptor for HA-mediated motility. In reviewing all the previous data on CD44, it's been implicated in cell motility, lymphocyte homing, that was how it was first described, and in tumorigenesis. Indeed, Paul Noble's group had shown that if you have CD44 knockout mice and you give them bleomycin, they have no problem in recruiting macrophages to the lung. They can't get rid of them. And they get exaggerated inflammation and significant mortality based on the fact that they can't resolve their inflammatory response. And we know that CD44 is also involved in the activation and expression of TGF-beta as well. For RHAMM, we know that we knew at that time that it promotes cell migration and proliferation. And in fact, TGF-beta 1 increases the expression of RHAMM. So there was a TGF-beta connection to this. We've subsequently shown that TLR2 activation in macrophages increases TGF-beta production and activity. So it's also a chicken and egg kind of system that we've been very interested in. And in other work, we showed that anti-RHAMM antibody can decrease the motility of macrophages after lung injury. So we looked at RHAMM then, since CD44 had already been looked at in terms of bleomycin injury, we wanted to look at what happened to RHAMM in this same model. So we looked initially at immunohistochemistry. This is the control slide where the antibody was mixed with the peptide that generated it. You can see that it's specific because there's no staining. This is diaminobenzidine staining with a methyl green counterstain. And you can see that the normal expression of RHAMM is in the epithelial cells, in the bronchial or smooth muscle, and in resident alveolar macrophages. I'm going to ignore this, which is a 2-hour time point where essentially the epithelial cells have died where the bleomycin was injured. So this could be non-specific staining. But when we looked at 4 and 7 days after, we now had this influx of macrophages. We're now highly positive for RHAMM. So were these the site of production of RHAMM, or macrophages that's engulfing RHAMM that was in injured or dead cells? So we wanted to look at in situ hybridization to figure out if this is the site of production. This is the sense control. This is the control saline with the antisense. And you can see that RHAMM is actually made in resident alveolar macrophages. And this is at 2 days after the injury, where you get the macrophages infiltrating, and they're highly positive for RHAMM mRNA. And that suggests that RHAMM is really is synthesized in the macrophages. And when we took the cells out of the animals and looked at their motility in vitro, the saline animals had an increased motility, suggesting that intratracheal saline is not a benign thing to do. We do this all the time to premature infants to supposedly clear their airway and their ventilators, and I sort of take pause and think, what the hell we're doing? But the bleomycin injured animals had a higher, macrophages from these animals had a higher motility, and both of these were completely abrogated in the presence of blocking RHAMM antibody, which we knew was a blocking antibody, but not with IgG, suggesting that RHAMM perhaps was going to be involved in macrophage recruitment. So in order to examine that, we actually gave anti-RHAMM antibody, massive quantities of this antibody, to rats that were bleomycin injured. So, and we looked at ED1 positive cells again. Bleomycin injured animals again showed us the increased ED1 positive cells accumulating in the lung. IgG had no effect, and we had about a 60-70% effect of blocking the macrophage influx with RHAMM antibody, which suggests that the interaction of RHAMM and HA, since we had the peptide stuff and now the RHAMM antibody, was important in the recruitment of macrophages to the lung after injury, which really is a fascinating thing if you think about it, where RHAMM HA is involved in recruitment of these cells, and HACD44 is involved in the resolution of inflammation. So the same ligand with two different receptors is doing vastly different things in the same model.
How was the antibody administered?
It was given IP, so systemic. Okay, so then Eva generated RHAMM knockout mice. We got the RHAMM knockout mice from her, and so we wondered if we could replicate these studies in the RHAMM knockout mice as a genetic way to think about it, because we were giving absolutely massive quantities of antibody to the rodents, so we wanted to make sure there was a genetic reason to do this. Okay. Okay, so the first thing we did was bleomycin is a great weight loss strategy, but I wouldn't recommend it.
I was going to say where you got
it. Because when you're really sick and you're expending a lot of energy just to breathe to stay alive, you lose a lot of weight in this system. So we measured the body weight percent of day 0 at the start. Wild type and knockout mice given either nothing or saline had increases in body weight, as you would expect. The wild type animals that got bleomycin intratrachely lost 15% of their body weight by days between 7 and 10, and then gradually increased their body weight as the respiratory distress got better. And the knockout mice, RHAMM knockout mice, were protected, so they didn't lose as much weight as the wild type animals. When we looked at the respiratory rates now with our fancy plethysmography system, we note that the wild type and knockout mice had no changes in respiratory rate from baseline. The wild type animals had early increases, which increased up to 10 days and then never quite got back down to normal, but were improving by the time we went to 28 days. The knockout mice were protected since they had lower respiratory rates than the wild type animals. When we looked at the macrophages, we now had an enzyme system called N-acetyl-glucosaminidase activity, which is an enzyme specific to N1 macrophages. We can measure this activity in the cell pellet that we get from bronchoalveolar lavage, and we could tell that the wild-type bleomycin animals, even at day 28, still had an inflammatory response that you could measure, but that the knockout animals actually had a significantly lower macrophage response. And then when we looked at the HA content of the lavage, we could tell that the wild-type animals, even as late as day 28, still had increased HA in the lung, whereas the knockout animals had a lot lower amount of HA. So what was the size of this HA? And maybe I didn't put that slide in. It was all overlapped with HA. So now we had demonstrated genetic deficiency of RHAMM had essentially what we would have predicted, which is decreased amounts of inflammation and decreased respiratory distress and decreased weight loss. So we wanted to do the counter experiment, which was overexpress RHAMM in macrophages and see if we could get the counter response. So we generated mice using the this promoter that was restricted to macrophages only, and we got this construct from Chris Glass in UCSD. It's actually a unique promoter because unlike lysem and lysem models, this is restricted entirely to macrophages. It does not get expressed in neutrophils, and it's a late macrophage marker, and that's scavenger receptor A, now that my long-term memory is returning. And we put a MIC tag, because we never use the hemagglutinin tag, which is HA, in the lab. So we used a MIC tag to identify the construct, and then we put the full-length cDNA for RHAMM into this construct. And we generated 4 transgenic lines, if you will, that were germline transmitters. So we could then assess in 4 separate lines whether the behavior of these animals and these cells were exactly the same in those 4 lines. So I'm going to spare you the enormous amount of data we generated to make sure that these animals were transgenic, that it was only expressed in the macrophages, that it resulted in cell surface expression of RHAMM, and it did not alter the expression of CD44 on the cell surface. So I'll spare you all that data and then bring you to bone marrow-derived macrophages, where we looked at chemotaxis of these macrophages to HA. And when you took wild-type animals, there was a approximately six-fold increase in the chemotaxis to HA in a Boyden chamber assay. And each one of the lines, there was a doubling or tripling of the response to HA in terms of chemotaxis with the animals that were expressing the RHAMM in macrophages. So then the question came up, is this just altering the environment, or is it really the effect of overexpression of RHAMM in macrophages that's causing this? And we racked our brains a lot as to try and how to figure this out, and my close friend and collaborator, Horace DeLisser, came up with this idea where you could give thioglycolate intraperitoneally, and in that model, the first cells that appear in the first day are neutrophils, and then further down the line, at 5 days, you get macrophages. So we wanted to look, after thioglycolate-induced peritonitis, what were the cells between the wild type and the transgenic lines that appeared both at 1 day and at later time points. So we first looked at neutrophils that appeared at 1 day after the thioglycolate installation. And in wild type animals, there was a huge influx of neutrophils into the peritoneum. And in fact, this was exactly the same in all the transgenic lines. So the neutrophil response was exactly the same in wild type and transgenic animals. When we looked earlier than 5 days, at 3 days, the wild type animals had not even started to respond with the macrophage response. And each one of the transgenic lines had a robust macrophage response to the peritonitis. So what that suggested to us is that it's not altering the environment of the milieu of it, but it was the expression of RHAMM within the macrophages that drove that inflammatory response. So naturally now, the next thing to do, of course, is to give them bleomycin and see what happens to their respiratory rates. And what we did was we compared the wild-type mice with 2 of the lines that were overexpressors, and controlled saline animals had no differences in respiratory rates. The wild-type animals, as we showed before, had increased respiratory rates that were getting better at 21 days. And both transgenic lines, 8 and 22, had increased respiratory rates as early as 4 days and never got better. And we had to actually stop the experiment at 21 days because these animals looked moribund and were losing a lot of weight. And the vet called us and sort of said, "What the hell did you do to these mice?" So we had to terminate the experiment at 21 days and not go on to 28 days. And when we looked at the HA content of these animals, the wild-type animals still had a little bit of HA like we showed before, but the transgenic line had a huge increase in HA. And so was this low molecular weight or high molecular weight? And when we look at control and saline, these is high molecular weight gels. These are fractionation gels that have been stained with alcid blue to demonstrate HA. We extracted HA and then ran it on the gel. And you can tell that the transgenic line really had low molecular weight HA as compared to the wild type. So what that suggested to us is that as our hypothesis was that if you overexpress RHAMM in the macrophages, you could now do the opposite of what the knockout showed us, which is increased inflammation, increased HA, and worse respiratory distress. So I'm going to sort of switch gears and talk about the other part of my life, which is bronchopulmonary dysplasia. So we have premature infants that are born, they're surfactant deficient, they have respiratory distress, we put them on ventilators. And you and I breathe by sucking air in, we create a negative pressure in our interpleural area, whereas this ventilator is really pushing air into the positive pressure. So it injures this fragile, underdeveloped lung and causes injury. So the model for what happens to babies' lungs is that we know, for example, there's a 5-stage process to creating a lung, from the lung bud through embryonic, pseudoglandular, canalicular, saccular, alveolar phases of lung development. And that what we're doing, in essence, by having preterm birth is interfering with intrauterine lung development. We know that lung development is plastic because you can give stressors so that cytokine exposures with chorioamnionitis, for example, or other stresses like placental insufficiency, etc., can alter the timeline progression of lung development. And well known since the 80s or 70s, when Liggins first published this, you can give antenatal corticosteroids, which enhances surfactant production and increases the maturity of the alveolar TAC2 cells to make surfactant, so they have less respiratory distress by about 50% when they're given antenatal steroids. But still, preterm birth exists. It's way too high. It's 11.4% of all births in the United States. And the nasty neonatologist comes and initiates ventilation. And now you are injuring the lung because you've got this positive pressure ventilation. The baby's lungs have to develop postnatally now, ex utero. And we are manifestly great at undernourishing these infants. We have not been able to reproduce what the placenta does. So we give them poor nutrition. I grew up in the era where we gave corticosteroids galore to try and get the inflammatory response controlled. We ended up knocking their IQs down by a significant portion in doing that. We don't do this anymore. And of course, the second hit of inflammation and cytokines happens with the exposure of oxygen, ventilation, and infection, which these babies are also prone to. And now you get the end result, which is bronchopulmonary dysplasia, where you get inhibition of alveolar and vascular development rather than fibrosis. So we wanted to study this, and remarkably, rodents are born at a lung development stage, which is in the saccular stage. And so they don't start alveolar development until day 3 of life. So their alveolar phase is entirely postnatal, from postnatal day 3 to about postnatal day 21, which gave us a beautiful window to try and study the alveolar development and how injury in the neonatal period might alter that. Peter Burry in the early 90s looked at normal lung development and showed this transition from the saccular to the alveolar phase, which is a remarkable, intricate endothelial cell invagination of the primary septi, which then creates alveoli to increase the gas exchange surface that we all need. And you can see the scanning electron microscopy that shows the complexity of this structure. We know that this is disrupted in preterm infants with BPD, but also in acute lung injury and people with emphysema and chronic obstructive pulmonary disease. And this is a sort of remarkable vascular X-ray, if you will, angiogram, where this is a term infant that died of non-respiratory illness at birth. So the normal development of the lung did occur, and you get this. This is the right pulmonary artery that's been injected with a radiopaque dye. You can see this arborization of the vascular tree. This is a preterm infant. It was born preterm but died at term equivalent age. And you can see how foreshortened the vascular tree is of this poor infant. This has been studied by the admin group in Colorado and others, including us. And when you antagonize VEGF receptor 2 by this compound called Sugen 5416 as a single shot at the time of birth, you essentially kill off all the endothelial cells, because VEGF is a critical factor for endothelial cell survival. And what you end up with is a simplified no alveolar development into adulthood. So this critical window of alveolarization is very critical. If you don't develop alveoli at that point, you never develop alveoli long term. So VEGF is really important in alveolar development. We wanted to look at HA and there's receptors. Low molecular weight HA had already been described to promote angiogenesis by West and Kumar and others. And so we wanted to see how the receptors were influencing endothelial cell behavior. And this is published work, so I'm going to sort of quickly just summarize it. And we used in vitro assays to determine which receptor was doing what. So we looked at adhesion to HA, proliferation, migration, and tube formation. And when we put anti-CD44 antibody, we got decreased adhesion to HA, decreased proliferation, but there was no effect on migration. When we used anti-RHAMM antibody, there was no effect on adhesion or proliferation, but there was a decrease in migration of endothelial cells. When you gave half maximal doses of the antibodies, you could both RHAMM and CD44 affected tube formation on matrigel. And then we then looked at these models in vivo angiogenesis, where we put a matrigel plug subcutaneously into these animals, put FGF or VEGF into these matrigel plugs, and looked at actually the hemoglobin of these plugs to look at the angiogenesis that was instilled. So without FGF, you got no promotion of angiogenesis into this matrigel plug. When you gave FGF with non-specific IgG, you got this nice, robust angiogenic response. CD44 almost had increased response. We later have found out that the integrity of the blood vessels in this model is affected, so that you do have angiogenesis defects in CD44 knockout mice. But when you gave anti-RHAMM antibody, you could completely aggregate this response, and you got no angiogenesis. So we're like, wow. And so we wanted to figure out what the mechanism was of this. We know, as I've told you, that VEGF is important in angiogenesis, and the way that it promotes angiogenesis is, and endothelial cell migration, is through the activation of endothelial nitric oxide synthase, or eNOS, and NO production. And so NO is important for angiogenesis, vascular remodeling, and wound healing. And so we looked at RHAMM knockout mice. One aspect that is controlled by angiogenesis that I've told you is alveolarization. And we looked at postnatal day 15 animals to look at their alveolar development. And you can do that by a morphometric mechanism called radial alveolar counts. So when we first looked at these wild type and knockout mice, either look at the saline, the wild type animals were exactly the same as saline. You can tell the wild type animals at day 15 had this nice, intricate, complex distal structure, and they had a normal radial alveolar count. The RHAMM knockout mice had less developed alveolarization at baseline. This is normal animals. And you could tell that their radial alveolar count was decreased in comparison to the wild type. We know from experiments that have been done that if you give exogenous VEGF to wild-type animals, you can increase alveolarization. And sure enough, in the wild-type animals, we could increase the alveolarization here, but there was no response in the knockout mice, suggesting that there was a signaling defect to VEGF in the RHAMM knockout mice. We had crossed these animals to FLIC-LAC-Z mice that express LAC-Z at the FLIC locus, so that as a heterozygous system, so you can now measure beta-galactosidase activity to get endothelial cell content. And sure enough, when we did that, the wild-type animals had increased content of endothelial cells by beta-galactosidase activity, and there was no effect in the RHAMM knockout mice. So since VEGF is on the pathway to make nitric oxide, then we said, okay, what if we gave nitric oxide back to these animals? Can you now rescue the knockout mice? So what we did was we had animals in either room air or 10 parts per million of nitric oxide for the duration of the experiment. And cut a long story short, room air animals had the same defect that I showed you already. There was a defect in RHAMM alveolarization, and this could be rescued with nitric oxide treatment, suggesting that it was really blocking the VEGF pathway to creating nitric oxide that was being affected. interestingly, when we looked at the beta-galactosidase activity, there was absolutely no difference in the content of endothelial cells, suggesting that this was really a functional defect and not a content defect, and suggested that it was the migration of the cells that was affected. We speculate that it was the migration of those endothelial cells that was affected by the lack of RHAMM. We wanted to look at an injury model that was an adult injury model to see if this was true, and that was purely an endothelial cell migration problem. So Phil Schall, who is my next door neighbor, who's the director of the Center for Pulmonary and Vascular Biology, amazing scientist, created this model where you could do an electrical injury to the carotid artery, and that just denudes the endothelial layer of the blood vessel, which over 4 to 7 days now those cells migrate and re-endothelialize. It's a model of acute vascular injury. If you then inject Evans blue dye into these mice, the denuded surface of the vasculature stains blue, whereas if you had endothelialization, the Evans blue dye does not stain. So blue dye means denuded surface area. So if you look at wild-type animals at day 7, you had almost complete resolution of the denuded area, whereas the RHAMM knockout mice still had denuded area at the same time, suggesting that there was a defect in repair or re-endothelialization of these animals and a defect in endothelial cell migration. So if this was really part of the nitric oxide pathway, giving a nitric oxide donor would ameliorate this. We gave mulsidomin, which is an in vivo NO donor that you can give, and you can actually completely rescue these mice from the denuded area. So that this is the area of the denuded area we could completely resolve back down to baseline in the knockout mice. So this sort of gave us the impetus to look at the pathway for nitric oxide production. So we wanted to move this into an in vitro system. And so we looked at single scratch wounding of endothelial cells that were made confluent. And when you give VEGF to these cells and you do scrambled siRNA, you get increased migration of these cells into the wound. But when we knocked down RHAMM, we had decreased migration. And this is quantified here where you get increase with VEGF. Scrambled peptide doesn't have, scrambled siRNA doesn't have any effect, but when you knock down RHAMM, you block the migration of these cells completely. So we now had an in vitro system by which we could analyze this whole thing. And if it's really on the pathway to nitric oxide production, we can now measure eNOS activity directly, which is a radioimmunoassay of the conversion of arginine to citrulline, and we did that with the help of Phil Schall. And you can tell that VEGF increases eNOS activity by 50%. Scrambled siRNA had no effect, and RHAMM siRNA could completely aggregate eNOS activity, suggesting that RHAMM really was blocking the pathway of VEGF to eNOS activation, which is amazing to us. So we wanted to reproduce the NO experiment, if you will, in vitro. If you give VEGF, you get increased migration. If you give the peptide that blocks eNOS or nitric oxide synthase, you can block the migration of these cells, confirming that VEGF is going through the nitric oxide pathway. But if you do siRNA to RHAMM, you don't get the migration. However, compared to spermin, if you give spermin nonoate, which is a steady NO donor over 18 hours, you can now rescue this migratory defect. So we could reproduce what we found in the mice in our in vitro system. So we were very happy about this. And that gave us the opportunity now to review what the pathway was for eNOS activation. So it's a fairly complex pathway. It happens in caveolae, and you get VEGF stimulation of the VEGFR2, you get phosphorylation at multiple sites in the VEGF receptor. You also get ERK phosphorylation, but you get then Sark activation, caveolin phosphorylation, PI3 kinase phosphorylation, AKT, eNOS phosphorylation at specific sites, and now eNOS dissociates from caveolin to produce nitric oxide, which then causes EC migration. So now this is sort of pathways that have been identified for a long time, and now we could define where in this pathway RHAMM was functioning. So we first did siRNA to RHAMM or scrambled. We got knockdown of RHAMM, and we looked at AKT and eNOS phosphorylation using phospho-specific antibodies. And we could tell that VEGF caused AKT phosphorylation, eNOS phosphorylation, and ERK phosphorylation. That the knockdown of RHAMM knocked out AKT, eNOS, but had no effect on ERK. So it was a specific effect on the AKT, eNOS pathway and not the ERK pathway. And this is quantified here by increasing AKT and eNOS, which was affected by knockdown of RHAMM and no effect on ERK. So we know that there's a number of different agonists that activate eNOS: HDL, insulin, estradiol, acetylcholine. So we wondered if this was a universal system. And to cut a long story short, HDL, which one is this? Insulin, estradiol, and acetylcholine had exactly the same thing, where we could knock down RHAMM for AKT and eNOS with no effect on ERK, suggesting that for every agonist that activates eNOS, RHAMM was an absolute requirement. Okay. So we think this is a universal system. Was it affecting the phosphorylation of the VEGF receptor? We took specific antibodies that are known to regulate migration, and there was no effect of RHAMM knockdown. And then if you pull down IP the entire VEGF-R2 and looked at phosphotyrosine, that wasn't affected either, suggesting that RHAMM was not involved in the phosphorylation of the receptor. Now, further upstream, Sark activation is pretty important in that first step after VEGF-R2. And Sark activation is a very complex system. You have a C-Sark kinase, CSK, that constitutively phosphorylates Y527 in Sark. And if that residue is phosphorylated, Sark is not active. When you get SHP2, which is a phosphatase, it dephosphorylates 527. Sark opens up, and then there's autophosphorylation of another tyrosine residue, 416. So Sark activation is decrease in 527 and increase in 416 phosphorylation. So no effect on VEGF-R2. You get decreased 527 and increased 416. When you knock down RHAMM, you now have hyperphosphorylation of 527 and no activation of 416. To note, phosphorylation of caveolin is downstream of this, so RHAMM is actually controlling the activation of Sark. So in summary and conclusion, RHAMM is critical to the inflammatory cell recruitment. It's necessary for eNOS activity to produce NO, endothelial cell migration regulated by NO, normal alveolarization and repair of endothelial cell injury is universally required for Sark activation to activate eNOS. It's an essential component of growth factor-mediated motility. And we speculate that RHAMM is going to be a potential therapeutic target to limit inflammation, prevent the effects of acute lung injury, but we're going to have to be cell-specific in our targeting, because if we block RHAMM, we're going to block alveolar health. We may stop inflammation, we may make alveolarization worse. Okay? I get to go around the country and the world giving these talks, but it's really these guys that do the work. I'm indebted to them for being incredible scientists that challenge me on a daily basis, and our collaborators Phil Shaw, Boris Loser, and Eva Turley, and our funding sources. Thank you.
So we have time for a few questions.
Very, very interesting presentation and excellent data. I have still a little bit of difficulty in understanding exactly how representative is the bleomycin-induced lung toxicity for acute respiratory distress syndrome, for acute lung injury, the way we define them histopathologically in humans, because we are looking for acute lung injury, we are looking for intraalveolar fibrin. For acute respiratory, adult respiratory distress syndrome, we are looking for formation of hyaline membranes in the exudative phase. So we have very specific things. Fibrosis, yes, it is known that bleomycin induces fibrosis as well in humans who receive bleomycin-based therapy. But that is a very long-term change that we see in lung. It's not an immediate thing that we see in the first 3 weeks. So in terms of histopathological changes in the mice who received bleomycin, what did you actually see in the first 2 to 3 weeks?
Right. So you're absolutely right. Models are models, and can only recapitulate some of the human condition. The reason we chose bleomycin was what I said, was that it's a model that's a survival model that goes through respiratory distress and then fibrosis. So hyaline membranes are actually an effect of surfactant deficiency. So early on, we didn't look histologically for hyaline membranes in that model. I would be surprised if they were not there. But the classical effect of bleomycin is really when cancer patients get bleomycin, it's a systemic application, so it's an endothelial injury, not an epithelial injury. And there you don't get an inflammatory response that's robust, but you get these fibroblast foci, very much in line with what you see in the pathology sections of IPF, right? So while we have studied subcutaneous dosing of bleomycin to mimic that, we really wanted a model that had an inflammatory response that we could study that resulted in fibrosis. So absolutely agree that this model is limited. It doesn't recapitulate ARDS, of course, but it is the model that is one of the only models that is a survivable model.
So what happens when you block when you use the blocking peptide? How that affects the hyaluronic acid production in the lung?
Yeah, so it's associated with less HA in the lung, and our speculation and hypothesis is that it's because it's decreasing the inflammatory response. So what the inflammatory cells or the macrophages are doing is they're jazzed up, they're making lots of TGF-beta and other cytokines that are increasing the production of HA in the stroma within the lung, fibroblasts in the lung. So that if you decrease that inflammatory response, you get less HA production. Jim?
Do the RHAMM null mice have unaffected macrophage development and the same number of circulating macrophages as wild type?
That's a brilliant question. We're actually in the process of doing the CBCs on them right now. We believe they're normal at baseline. And when you look at M1, M2 differentiation of the macrophages, they essentially don't recruit M1 macrophages.
Okay. Are there other questions? Oh, one more.
Have you used this, or you use this model to study other, like a wound injury or other type of injury, like a infarction, to see how that affects the...
Great question. So we've done that in the CD44 knockouts. We're going to do that in the RHAMM knockouts. My prediction would be that there would be a delay in the wound healing response because your angiogenesis is going to be affected, your epithelial regeneration is going to be affected probably, and the inflammatory response is going to be affected.
And those RHAMM null mice have defective excisional wound repair.
Oh, that's right. I'm sorry. Evatorly has done those studies. Actually, I forget. I started those when I was in Irvine. Long-term memory going. So they do have defects in excisional wounds. That's correct.
One last question.
How does cell surface RHAMM signal without a transmembrane?
That's the killer question. Right. I'm going to go balls with the scratching of my head on that one. So RHAMM is on the surface. That's no question about that. How it gets to the surface is really an enigma. It does not have a signal peptide. It does not have a transmembrane domain. And yet multiple flow cytometry, antibody binding, biotin labeling, a ton of biotin-limited HA studies, etc., antibody blockade, demonstrate that this is on the cell surface. It's got a CD name, which is hard to get. It's CD168, which recognizes cell surface proteins. And yet, how does it do that? At one point, we thought it might be a GPI-anchored protein, but I don't think that's correct. FGF, of course, shares this unique trait in that they don't have signal peptides or transmembrane domains, but nobody argues that they're on the cell surface. So that's the first piece. The second piece is, how does it signal this? My sense is that there's going to need some cooperative transmembrane protein, either a GPI-anchored protein or a transmembrane protein that promotes that signaling. That still has to be worked out. We know that if you give HA6 in this system, in vitro, you can activate the eNOS pathway. I didn't give you that data in today, but you can activate eNOS activity by giving low molecular weight oligo HA.
Okay, thank you so much.